Okay. So today is the second talk in our series on gene therapy, honoring Ken Burns, our friend and our leader. And our speaker today is Dr. Shannon Boye. Shannon Boye, who calls herself Ken's science granddaughter. And I didn't know why, but I can figure that out because she got her PhD the year I retired from UF. So you can see how young she is. She's now a professor and chief of the Division of Cellular and Molecular Therapy in the Department of Pediatrics here at UF. The focus of her research is developing viral vector-based gene therapies for the treatment of certain ocular diseases. She has multiple awarded and pending patents emanating from her research, and she's been actively funded by the NIH, by private foundations, and also by Big Pharma. She's the recipient of several major awards, including the Foundation for Fighting Blindness, their Board of Directors Award. The Foundation for Fighting Blindness, the Board of Directors Award, which is the Gund Harrington Scholar Award. She received the Arvo Foundation Merck Innovative Ophthalmology Research Award and the Arvo Pfizer Carl Thomas Translational Research Award. She's the co-founder and CSO, which I guess stands for Chief Scientific Officer. Yeah, okay. The CSO of, I need my eyes tested, of Alcina Therapeutics, which is a clinical stage gene therapy company. She's passionate about patient outreach and routinely provides lab hours for visually impaired patients, hosts foundation meetings, and educates patients about ongoing research being conducted to address treatments for their conditions. So please welcome our speaker today, Dr. Shannon Boye.
Thank you, Pushpa. Can everyone hear me okay? Okay, great. So I come to you today as Shannon Boye, but also as Ken Burns' science granddaughter to honor his memory in this very special seminar series. I was very fortunate to come from a lineage of people that were experts in adeno-associated virus. So Ken mentored my postdoctoral mentor, Bill Houseworth, both of whom are giants in the field. And I learned so much from them. And I have my career to, I owe my career to them and their prowess in this space, really. So I'm really happy to be here. Thank you for the invitation, Arun and Pushpa. So I thought I'd start with just an introduction to me and why I got into science, and then take you through kind of a highlight of my career thus far. So it's weird to start any talk with the next picture that I can't show. Oh. There we go. Okay. What does O.J. Simpson have to do with my science career? So when I was in high school in upstate New York, I went to Our Lady of Mercy. It was an all-girls Catholic school, and we had highly sophisticated science equipment, as you can see. But this was my AP biology course, and it was taking place right around the time that O.J. Simpson was, on trial for murder. And my teacher used that experience to teach us about DNA fingerprinting. And unfortunately, the blood at the crime scene, at O.J.'s crime scene, well, maybe it wasn't his, but we know it was not handled properly. It was mishandled, so they could never actually do DNA analysis on what was found at the crime scene. But she taught us this awesome method where you could use restriction enzymes to cut up DNA to tie the DNA at the scene to the person that convicted the crime. And I thought that was the coolest thing in the world, that DNA couldn't lie. So that was sort of how O.J. Simpson got me really interested in becoming a scientist, if that's possible. So as I mentioned, I grew up in upstate New York, and these are not real pictures of my childhood, but they could be. I was very cold all the time. So then I entered what I like to call my marine biologist phase, and I was just talking with Dr. Brockman here. My husband and I always say that scientists are all just really, frustrated marine biologists. No matter what field of science you go into, excuse me, you started as someone that loved marine biology. So I wanted to move from this cold weather to go and scuba dive and study tropical things for the rest of my life. I got a little bit wiser as the years went on, and I went to do an internship at the Whitney Marine Lab, which is an offshoot of the University of Florida that's located just south of St. Augustine. And I was very much attracted to this internship based solely on the overhead photo. It looked beautiful and warm. But this wound up being really pivotal in my life because it was a place where they married marine biology with biomedical research. And so it got me, pushed me more in the direction of working on human disease and biomedical research. And my mentor at the time there, Paul Linzer, he suggested that I go to graduate school, I get my PhD. And so he came and took me on a tour of the University of Florida, and I fell in love with it. So I came down here in 2001 to get my PhD. And I did that in 2006 in the Department of Neuroscience. I then married another gene therapist, which has also been sort of a pivotal moment in my life and career because we work together. We see each other all day, all night. We still get along. But we have very different skill sets, and I think we complement each other, and that's made my work life a lot easier, honestly. I owe a lot to Sanford. So as Pushpa mentioned, I'm now a professor in the Department of Pediatrics at UF with my own independent lab. And I've also co-founded a company called Atsena Therapeutics, for which I serve as CSO. And so it's been amazing for me to have my toe dipped in both the academic life as well as the industry life, because they're very, very different. But I like them both for different reasons. But the company has really been the vessel by which I've taken my academic research and been able to apply it to patients. So it's been a really, really exciting ride. Okay, so a few things that I wanted to cover today. I'm going to keep this fairly high level, so I wanted to give some background so everyone understands the subsequent slides. I'm sure that Arun gave an excellent introduction to this last week, but we'll start with a very high level, what is gene therapy explanation. And then for those of you in the audience that don't study the eye every day, I'll do a little bit of a brief ocular anatomy lecture. And then we'll talk about ways that we evaluate the health of the retina. So how do we know if the retinal structure or the retinal function is good? And you can do this in both patients in an ophthalmology clinic or in mice in your research lab. And then I'm going to convince you why I hope the eye is the most amenable organ for treatment with gene therapy. And then finally, excitingly, I'm going to give you an example of something that we've taken from bench to bedside and show you what we see in those patients in real time. Okay, so all of us in the room are made up of DNA, and within that DNA are these little subunits called genes. My genes are different from your genes. That's why you have blue eyes, I have brown eyes. But genes do something important, and that's that they make proteins. And proteins you can think of like the building blocks of life. They act alone or in conjunction with other proteins every second of the day and night to perform essential functions in our body. So genes make proteins, proteins perform functions. Unfortunately, sometimes we can have misspellings in our genes, and then the result of that is either that gene doesn't make the protein it was supposed to make, or maybe it makes it, but it's in a really funky shape and it can't go on and interact with its binding partners. Either way, the end result is that function is not performed. So that really forms the basis for gene therapy and what it is at a high level. We're asking the question, can we take DNA that encodes a normal gene and package it into a vector and deliver that vector to the target cell? Once inside that cell, that vector will release that normal gene. That normal gene can go on to make that normal protein it was supposed to produce and hopefully restore function to those cells. So how do we do it? What's this vector? That's where Ken comes in, because he's really the grandfather of a very important, what we call, I guess Arun calls it the awesome virus now, adeno-associated virus, or AAV. Why is it awesome? Because it's the gold standard. It's being used in hundreds of clinical trials worldwide now to treat human disease. It's a non-pathogenic virus. It doesn't make you sick. It's relatively non-immunogenic, especially when it's placed in the eye. And what it does is it drives efficient, sustained expression of the genes that it's delivering. So it drops off the gene, and that gene expresses for the lifetime of that living cell. And again, as I mentioned, it's clinically relevant. It's being used right now to treat human conditions. Excuse me. Okay, a little bit on retinal anatomy. What you're looking at is a cross-section of the human eyeball. And at the very back of that is this tiny little tissue, which is in red here, called the retina. Despite its small size, it's very complex. There are multiple layers of neurons in this retina and glial cells. So when light comes into the eye, it has to go through your cornea, through that vitreous jelly in the middle of the globe, and then it actually has to cross over all of those retinal layers until it lands on the photoreceptors, which are on the very back of the retina. And that's where vision really begins. That's where photons are converted into an electrochemical signal that's sent to the brain and processed as vision. So we have two different types of photoreceptors. And as these cute little cartoons tell you, we use them under very different lighting conditions. So our rod photoreceptors are what we use under dim light conditions. So you wake up in the middle of the night and you're trying to navigate to the restroom, you'd be using your rods. Our cones, on the other hand, are what we use for high acuity color vision. They're in the center of your retina, which I'll talk about in a moment, but that's what you're looking at the screen with right now, is your very central cone photoreceptors. In the primate retina, the rods and the cones are situated in a very specific way. So this on the right is what's called a fundus image. It's simply a picture of the back of the retina. And you can see in this hyperpigmented region here, which we call the macula, that that's where the cones are concentrated. And at the very center of that macula is a pinhole-sized structure called the fovea, which is a cone-exclusive region of the retina. Your rod photoreceptors, on the other hand, are concentrated further into the peripheral part of your retina. So again, it makes sense that you're looking at the screen right now with your central retina because you're using those cones for high visual acuity. And then rods being in the periphery, I like to give this example. If you've ever been camping and you've looked up at a star, you might notice that you can't see it as well if you look straight at it. But if you kind of look at it out of the corner of your eye, it gets a little bit brighter. And that's because your rods are more sensitive and can detect lower levels of photons. This is just a cross section, if you're interested, through the foveal pit of a monkey. And you can see in this region of the retina, these are the photoreceptors here. And all of the other neurons have essentially moved out of the way, which allows light the most direct path to those photoreceptors, which are constantly signaling and providing us with that high visual acuity. Okay, so where do these signals go? They start in the photoreceptors, and through glutamate signaling and a bunch of other neurotransmitters, they're sent downstream to the ganglion cells, and then ultimately through their axons, which all converge on the optic nerve head. This is ganglion cell axons in a flat mount of a retina. And then from there, they move to the brain, to various regions of the brain where they're processed for higher order vision. Okay, so that's retinal anatomy 101. Now, in order to understand the clinical trial results that I'll speak about later, I just want to go over some of the outcome measures that are used to analyze retinal structure and function in people and in lab animals. So again, I showed this before, but this is a normal looking patient fundus image. In this fundus image, you can see an example of a patient that has had a retinal detachment, unfortunately, right over that cone-exclusive fovea. So this patient will have experienced a sudden vision loss. But that's one way a fundus image can give you an idea of what's happening with retinal health. This patient has a disease called age-related macular degeneration, which is characterized by the buildup of these fluorescent deposits that start in the macula. And then on the flip side, this is a patient that has a disease called retinitis pigmentosa, which, as its name implies, is characterized by this pigment deposition. And this starts in the peripheral retina and ultimately encroaches on the central retina until the patient develops tunnel vision and ultimately blindness. So fundus imaging is kind of a quick and dirty way of looking at retinal structure, but a new technique emerged a number of years ago that has really advanced the field, and that's called optical coherence tomography. So here's the en-face fundus image, and up above, you can see this OCT scan gives you a cross-sectional view of what's going on in the retina. And this has revolutionized the field, really, because now we can look at the retina in the patient or in the mouse over periods of time in life. We don't have to sacrifice the mouse and look at retinal histology. We can do this in life over time multiple times. And we know by corresponding these scans to histological sections what each of those layers refers to. So you don't have to memorize all of this, but another thing I'll say is that you can think of this like an ultrasound of the eyeball, only instead of using sound, we're using light that bounces off those layers in different ways. So this is an example of an OCT scan that shows that there's a problem with this patient's retina. So again, this patient has experienced a detachment right over the fovea. So this patient would have immediately lost their central vision. But this is a very powerful tool for looking at retinal structure. What about retinal function? So you've all probably heard of an EKG before. That's a non-invasive measure of heart health. Similarly, an ERG, or an electroretinogram, is a non-invasive measure of retinal health. And it's based on the concept that current flows from a source to a sink. So the current that's being generated in your retina actually flows out through your corneal surface and can be picked up there by placing an electrode on your cornea. And these come in a variety of really uncomfortable looking shapes and sizes. This is a difficult outcome measure to perform on children because it's not very comfortable, but this can be done in a number of ways. But essentially, the electrode is placed on the corneal surface. The patient places their head inside a dome, and then flashes of light are presented to the patient. And then the computer reads out a waveform that looks something like this. So the first wave here, this is a negative going hyperpolarization of the photoreceptors in response to light. And then you have a positive going depolarization of the inner retinal neurons in response to the photoreceptor impulse. Okay, so ERG is one of the classic ways to measure retinal function. Another way to look at sensitivity of the retina is sort of a newer outcome measure called full field stimulus threshold testing, or FST for short. This is an outcome measure that can only be done on patients because it requires input. So the patient puts their head inside a dome similar to the ERG. They're presented with flashes of light, and those flashes of light are randomized in terms of their intensity. And then the patient, if they can see the flash, hits a button and says, yes, I saw it, or does not click the button because they didn't see it. So it essentially is measuring what's the lowest level of light that that person can see. And again, it's a measure of retinal sensitivity. There are also visually guided behavior tests that can be done. This one, of course, is only being done on a patient because you can't ask a mouse, for instance, to go through a maze unless you enhance or entice it with cheese or some kind of treat. But this is better done on human patients. But this is called the MLMT maze, and it was developed in one of the gene therapies that ultimately went on to be the first approved ocular gene therapy in the world. And basically, it measures the patient's visually guided behavior by the amount of time it takes them to find the exit, as well as how many times they bump into the objects in the middle of the maze. So you can actually do a version of this in your lab mouse. We call this the Morris water maze. And what we do is we subject the mouse, we dunk them in this pool of water. They can swim, don't worry. And they can see this platform that demarcates the slightly submerged, or sorry, they can see the flag that demarcates the slightly submerged platform. And if they can see, they'll swim right to it. If they can't, they'll sort of swim around. And I'll show you an example of that in a moment. There's another behavior test we can do on mice called optokinetic reflex testing. And I like to describe this as if you can imagine driving in a car by evenly spaced trees, you kind of get this feeling of doing this, right? It's sort of a subconscious response to visual stimuli. And mice will actually do this. You place them on a platform surrounded by this drum of rotating black and white stripes, and then they will reflexively move their head in the direction of the pattern of rotation. Then, of course, everyone is familiar with measuring visual acuity via an eye chart. We've all seen the big E before. Some more complicated outcome measures that I won't talk about today, but you can use pupillometry. So that's where you measure pupil constriction in response to a light stimulus. And if a patient is visually impaired, their pupils might not constrict as effectively. And then at a really high level, you can use fMRI, or functional magnetic resonance imaging, to look at how the visual cortex actually responds to visual stimulus, which is pretty high tech. Okay, so there's retinal anatomy and how to evaluate the health of the retina. So now I think I can easily convince you that the eye is the best place in all the body to do gene therapy. And Arun and I talk about this all the time. He always says I chose well, that I went into the eye, and really it was I got very lucky. I landed in a lab in the eye. But why is the eye so good for gene therapy? It's because it's small in size and you don't need a lot of vector to get the job done. Making vector is really, really expensive. Clinical vector is extremely expensive. So just to put this in perspective, in the Duchenne's muscular dystrophy trial that Sarepta ran, they used 5 × 10 to the 15th vector genomes in each patient. In the LCA1 trial that I'm going to talk to you about today, we use 1 × 10 to the 11th. So quite a big difference. So you don't need a lot to get the job done. It's also compartmentalized. So what you put inside the eye, for the most part, doesn't get out, which is good from a safety standpoint. The eye is also immune privileged. We have a few organs or tissues in our body that have evolved to be immune privileged. So for instance, testes, the eye, they're not going to respond in the same way to an infection or an antigen as your skin would. Otherwise, we wouldn't be able to procreate or see. So there's an evolutionary advantage to doing gene therapy in an immune privileged organ like the eye. And then finally, you always have a contralateral control, which, you don't have two hearts, but you have two eyes. So it's nice in clinical trials to be able to treat your right eye and follow progression in that eye compared to the untreated eye. That's always good to have a control when you're a scientist. Okay, so the disease we're going to cover today is a specific form of Leber congenital amaurosis caused by mutations in a gene called GUCY2D or Gucci2D. So LCA is actually a family of inherited retinal diseases that together account for the most severe form of pediatric retinal dystrophy. But there are about 20 different genes that, when mutated, can lead to one form of LCA or the other. Gucci2D was the first gene found to be associated with LCA, and it's one of the predominant forms. So LCA1 typically presents within the first few months of life. Moms and dads notice that their infant children aren't looking at them directly, or they're not fixating on an object. A lot of these babies have a roving eye movement called an nystagmus. So they're diagnosed relatively quickly, early in age, with very profound visual impairment, if not total blindness. There is a range, but it's profound. And the mechanism underlying this disease is extremely clear, and I'll get into that in more detail on the next slide. But this gene encodes a protein called retinal guanylate cyclase 1. Remember, genes make proteins, proteins perform functions. And the function of this protein is to participate in phototransduction. So a process where we convert a photon of light into that signal that's sent to the brain. GC1 is playing a role in that process. So without functional GC1, that phototransduction cascade shuts down. And I'll show you how that is right now. But then, just as a final statement, this is a protein expressed exclusively in the outer segments of the photoreceptor cells of the retina. Okay, so this is a simplified it's not really simple, sorry. You don't have to remember any of this, but when a photon of light hits the eye, it interacts with an opsin molecule and it sets off this cascade of events. As a result, these cyclic nucleotide gated channels in the photoreceptor cell membranes close and calcium levels drop. When that calcium level drops, it comes unbound from this little protein called guanylate cyclase activating protein. That in turn activates guanylate cyclase, whose job is to make cyclic GMP, rebind, and reopen those channels. So it's not that complicated, I promise. If you don't have guanylate cyclase, you can never reopen these channels and get calcium back into the cell. So the photoreceptors in these patients are stuck in this chronically light adapted state. Fortunately for these patients, though, despite the profound dysfunction in their retina, they have excellent retinal structure, which for a gene therapist is sort of a dream, because that means that the structure is there for us to deliver that healthy gene back to, right? If we didn't have any photoreceptors in these patients, gene therapy wouldn't be possible. But this is a study that was done by a clinical colleague of mine who used OCT. Remember, that's that imaging technology, ultrasound of the eyeball, to look at a normal patient's retina compared to some of his LCA1 patient retinas. And he's color-coded different regions of those retinas with, that are important for different reasons, which we won't get into today. But on the bottom here really tells you the bottom line, and that's that he's plotted the thickness of those layers and put them against that normal range, which you see in light blue or in yellow. And you'll see that the vast majority of these patients have totally normal retinal structure. So this is really, really good news for them. Despite being profoundly visually impaired, their photoreceptors remain over their lifetime and perhaps therefore make them amenable to gene replacement therapy. In terms of their function, though, you'll see it's quite a different story. These are ERG traces. So on the far left, you're looking at traces from a normal patient. These are rod photoreceptor responses, and these are cone photoreceptor responses. You can see that a lot of the LCA1 patients do have some rod function, but it's really abnormal, whereas none of the patients that he evaluated had any recordable cone function. So this is a disease of profound cone dysfunction, abnormal rod dysfunction, but relatively great preservation of retinal structure. So when I started as a graduate student in the University of Florida, I was evaluating a chicken model of this disease. It was this naturally occurring model of LCA1. I don't miss those days because chickens were kind of hard to work with, if I'm telling the truth. So I moved very quickly into studying mammalian models of this disease, and that included the GC1 single knockout mouse, as well as a GC1, GC2 double knockout mouse. Just like the patients, the GC1 knockout mouse has totally unrecordable cone ERGs. The cones degenerate, and the rod ERGs are sort of abnormal. But this is probably the closest model of the disease that we have. Why do the rods stick around in this mouse? It's because of a very close relative of GC1 called GC2. And we know that because when we knock it out on top of GC1 in the double knockout mouse, we have a situation where both rods and cones are dysfunctional. So we've tested our gene therapy in both of these mouse models. This is just a smattering of papers that we published over the years. But the high level summary of all of those studies taken together are that when we treated these mice with subretinally injected AAV, and I should mention, this is how we get the virus to the photoreceptors in mice. So this is a surgery. It's done in mice and in people in the clinic, but it involves temporary detachment of the retina from the underlying epithelial cell layer. But what that does is it allows that vector immediate access to the photoreceptors, which are, remember, right here at the very back of the retina. So we treated these mice with our AAV vector. It contained a special regulatory region that restricted expression of the therapeutic gene to those photoreceptors. And then even 1 year after treatment, when we look at the ERG, we can see in the treated eyes, which are the blue lines, that there are significantly improved rod responses and significantly improved cone responses in those treated mice. And in red are the responses from the untreated eye. So therein lies the beauty of having a contralateral control. So clearly improving retinal function in these mice. We looked at the retinal sections of those treated mice and we probed them with an antibody directed against GC1. So you can see that stained for in green. These are two of our GC1 knockout mice. These are their treated eyes. These are their untreated eyes. And here's a wild type mouse for reference. So you can see that GC1 is expressing in the photoreceptor outer segments just where it's supposed to be. And we're not seeing any off-target expression. Okay, on this slide, let me just give a brief intro. So I made this video when I was a postdoc, so it's pretty cheeky. It's been a long time since I made this video. It was right after iMovie came out, so I was a little bit of a goofball when I made it. But this was a movie looking at visually guided behavior testing in our mouse models. So we took a normal mouse, an untreated GC1 knockout mouse, and then a GC1 knockout mouse that was treated with our AAV vector. And they're all age matched, and this is 1 year post-treatment. And we subjected them to the Morris water maze, which I showed you a picture of before. That was my technician. Okay, here's our wild type mouse, and we're measuring their visual behavior and the amount of it takes them to escape to the platform. So he goes right there, totally sighted. The next mouse is our untreated GC1 knockout mouse at 1 year of age. He was also our lazy mouse. I'm not kidding. We had to like poke him with a stick to make him swim. He had a personality. I'm kidding. But you can see that this mouse clearly has no idea that the platform is there. He doesn't see the flag. He's swimming into the wall like he doesn't know that it's there. We do eventually take the mouse out at 60 seconds for any reason. But he almost gets there and he doesn't. Clearly this is visually impaired. We randomize the platform and the release location so they can't remember how to do this right away. So yes, that's our blind mouse. And then this is our AAV treated mouse at 1 year of age, so 11 months post-treatment. So clearly, I mean, all of these gene therapy experiments were worth it. We restored vision to mice, but I didn't get into the field to restore vision to blind mice. I want to do this in people. So that became the next goal. So around 2014, we partnered with a company called Genzyme, and they were a company at that time focused on developing treatments for rare disease, and they were interested in this program. So my husband and I had covered the technology through UF, and they bought the technology, and we set off on a path with Genzyme Sanofi to do what we call IND enabling studies. So none of this cute, mice swimming around and me taking cute notes in my lab notebook. It was highly documented, very important big pharma level IND enabling stuff. But on this slide is kind of a summary of all of that. And I don't mean to bore you, but it's really very simple. On the left are all of the doses that we tested in our mouse models. And the second column is whether or not we saw efficacy at that dose or if we saw some toxicity. You'll note that once we get up to too high of a dose, we start to see toxicity. So there is a window here. We did another round of experiments looking at transduction in monkey. So we would give each dose of the vector and then count how many photoreceptors were expressing the gene at that dose. So we could correlate that percent photoreceptor transduction to efficacy to dose. And then the final study that we did is what's done before any clinical trial starts, which is a GLP safety study and toxicology study. So that's where you take your drug and you typically put it in monkeys and you make sure that it's safe. And so it involves like high level pathology and making absolutely sure with a bunch of clinical assessments that it's safe to go into people. And so with that study, we identified what's called our NOAEL, which stands for no observable adverse effect level. So we know that this is the dose that we can't go over in patients. So this is really 2 decades in one slide because I started this work in 2004. It's 20 years since then, but we've done a lot of work in these preclinical models. There are scientists that, of course, that dedicated their lives to understanding this disease in patients. So characterizing these LCO1 patients and, contacting me and saying, these patients look really good. They're good candidates for gene replacement therapy. In 2014, 2015 is when I aligned with Genzyme, which was almost immediately acquired by Sanofi. So that was a fun transition, but a great team of people that helped me complete all of the IND enabling work. And then where we are today, we've fully enrolled our phase 1/2 clinical trial, which I'll summarize in a minute. And we've received a number of regulatory designations like orphan drug designation, rare pediatric disease, and RMAT. And some of these are really useful in terms of getting extra access to the FDA to help expedite our path forward. Okay, so for the next few minutes, I'm just going to go over the results of our phase 1/2 clinical trial. And I know Pushpa told me no graphs or tables, but there is a little bit of patient data and some graphs, but I'll walk you through it. Okay, so what is the drug that we're delivering? What is the gene therapy? We're calling it at CINA ATSN 101, which is an AAV5 vector containing the human Gucci 2D gene being driven by a photoreceptor specific promoter to restrict expression of that gene to those cells. So this is just a picture of what that looks like, and it's being delivered by subretinal injection. Okay, this is simply the trial design. So what I'm showing you up top is the design of our dose escalation phase. So we had 3 different cohorts of patients and 3 patients in each cohort that received either a low, a mid, or a high dose of the drug. And then from there, we went into an expansion phase wherein we treated 3 adults with the highest dose and then 3 pediatric patients with that highest dose. So in order to be eligible for enrollment in this trial, you needed to have, structure remaining that was visible on OCT. You had to have, obviously, mutations in the gene that were confirmed with genetic testing. In the part A, we wanted patients to have BCVA of 2200 or worse, and we lightened that up a little bit in part B, where they had to have 2080 or worse. So the primary endpoint in this trial, like all phase 1/2 clinical trials, was safety. And the secondary endpoints were BCVA, so looking at that line, the eye chart, full field stimulus testing, which was that measure of retinal sensitivity we talked about, the multiluminance mobility test, which is the maze we talked about, and then we always give these patients visual function questionnaires, which are related to activities of daily living, quality of life, just how do you feel post-treatment, that kind of thing. These are just some baseline characteristics of those patients. Again, just a lot of numbers, but I think the take-home message here is in cohorts 1, 2, and 3, we had an age range of 18 to 44. In our last 2 cohorts, we had an age range of 12 all the way up to 76. So one of the coolest things in this trial, I think, is seeing functional improvements in a 76-year-old. We had a range of entry-level BCVAs, sort of all over the place, but again, these patients are all profoundly visually impaired. Okay, so first, safety. we were very excited to report that we had no serious adverse events tied to the medicine itself. So you always deliver this via subretinal surgery, and with all subretinal surgeries does come some risk of a surgical complication. So for instance, we had one SAE where a patient developed endophthalmitis, which is an infection in the eye, but that didn't have to do with the gene therapy, it had to do with the surgery. But we're very happy that we had no adverse events related to the gene therapy itself. We did have some patients that had a little bit of ocular inflammation. Again, this is very typical in ocular gene therapy, but it was transient and reversible with a quick round of steroids. And in the patients that did have inflammation, we still were able to see functional improvements. Okay, this is simple, I promise. All right, on what you're looking at here are our FST results in these patients. So on the x-axis, you have days post-treatment. And on the y-axis, you have a change in FST. So if your line goes up, that means your retinal sensitivity is improving. So in red, you can see the treated eye, and in black, you can see the untreated eye. And this test was done with a white light stimulus, a blue light stimulus, or a red light stimulus. So you can see that with all three colors, we had significant improvements in retinal sensitivity in our high-dose patients. So in one patient, the average, I think, increase was about 100-fold improvement in retinal sensitivity, but we had one patient that had a 10,000-fold improvement in retinal sensitivity. And that's akin to someone being able at first to only operate under, in a room that had bright sunlight and going to being in a room that was lit by the full moon and being able to navigate. So that's a huge improvement in the sensitivity of that patient's retina. This is also FST data simply showing you a dose response. So in red is the high dose, in black is the mid dose, and in blue is the low dose. You can see clearly that the patients that got the highest dose vector responded the best to treatment. This is our maze data. So in this maze, you are asked to navigate something that looks like this up top at extremely bright levels of light going down to extremely low levels of light. And so what we saw in this, we didn't enroll all of our patients in this outcome measure because we took the trial over at Atsena sort of a quarter of the way through. We got it back from Sanofi. But the FDA is okay with this outcome measure in terms of they use it to approve drugs, but they want to see is a two-step improvement in the patient's behavior. And what we saw is a two-step improvement in most of our treated patients, with most of them approaching a perfect score at the point that they had all been measured. So what this tells us is that these patients are also exhibiting the ability to navigate in real life. This is a real life improvement in their vision. In terms of BCVA, again, this is looking at the eye chart. The FDA sets an extremely stringent threshold on what they require to see to be determined clinically significant. So they want a patient to experience an improvement of 3 lines of vision. And for anyone that's looked at an eye chart, I think you would all agree that 3 lines is an amazing improvement. I would benefit from 1 line of improvement, let alone 3. And I think that that's changing. The regulatory landscape seems to be changing a little bit. But what we achieved in our high dose patients was around a 2-line improvement. But still, I think, again, if you've looked at an eye chart, you would all agree that that's an appreciable difference in vision. And then here are the patient-reported outcomes. Again, these are just questionnaires given to the patients about their activities of daily living, and you can see that over time, their scores improved. So they are perceiving a real change in the way that they navigate through their daily life as a result of this treatment. Okay, so those were all kind of hard, cold numbers and graphs, but I love this video. This was a video of a little girl named Aiden who had received the treatment. And then when she went outside on this day, it was snowing and she could see snowflakes for the first time. And her mom fortunately had her iPhone handy and was able to capture the moment.
Is that snow?
Yeah.
What did you just tell me?
I saw like the big snowflakes.
You could
coming
see
down.
some of the big snowflakes?
Yeah.
That's so cool, Aiden.
Yeah, I can see them right now.
You can see them right now?
Yeah.
There's a big snowflake in your hair.
Oh, yeah.
So yeah, very cool. So in conclusion, that was a very, very rapid run through this clinical trial and everything that went into it. But we've shown that this gene therapy is safe. We have no drug-related SAEs as of 12 months post-treatment, and we've taken that out further now, and the same remains true. Ocular inflammation was infrequent and correctable with steroids. And then in terms of efficacy, we are seeing significant and clinically meaningful improvements in retinal sensitivity on FST and approvable levels of improvement in visually guided behavior using a maze test that the FDA uses to approve some of these gene therapies. And we were super excited. This was like a bucket list thing, but we published those results in the Lancet just a few weeks ago. So with that, I want to thank everybody that has ever been in my lab. I've been at UF for a while now, and they've all sort of been on this journey with me. So I thank them. Everyone at Genzyme and Sanofi, who again was responsible for the IND enabling aspects of this work. The project then transferred to Atsena Therapeutics, so which I co-founded with my husband, who's on the left of me on the top right. And that's our CEO and our CMO. And I love these people. They're all in it for really good reasons. And then finally, I want to thank two important people. One on the bottom left, who was one of my favorite people in the world, Sam Jacobson, who Bill and Ken both worked with as well. But he was just the ultimate mentor, and he dedicated his life to treating these patients. And he passed away about 1.5 years ago. And then finally, in loving memory of Ken, who I was telling his wife that we didn't work together a lot, but every time I was with him, I was just astounded by his intelligence. He was like the smartest man, and he intimidated me a lot. But I'm so fortunate to have come from a long line of brilliant men who knew everything about AAV. And without their mentorship, I wouldn't be doing what I'm doing today and using the tool that they worked on so hard to help actual people. So owe a lot to Ken. Thank you for putting this whole symposium together. It was an awesome memory of Ken.
Questions? Is this on? Questions? Okay, let's start here.
You mentioned that you are looking at time periods beyond 12 months. Does it look like there's continued improvement of receptor function, or does it plateau?
So the treatment effect shows up around 1 month. Sorry. The treatment effect shows up around 1 month post-treatment. It crawls up a little bit until about 3 months post-treatment, and then it plateaus. And we've now followed our first treated patient out to almost 5 years. He was treated on November 19th. It'll be the 5-year anniversary in a few weeks, and they're plateaued. It's totally stable.
Wonderful.
Do all of the patients have same genetic defect, same base changes, or does that matter?
No, they don't all have the same base changes, but what they're required to have is biallelic recessive mutations. So they get one bad copy from mom, one bad copy from dad, but they're not all the same.
And I missed it. Was that the rhodopsin protein that there was defective in these patients?
No, guanylate cyclase.
Oh, the cyclase.
Mm-hmm. Yeah, you're welcome.
Question on chat. Pam, would you get that? Acuity obtained with treatment.
So I would have to have the paper in front of me to remember, but I do know that the biggest improvement we saw was 2 lines. So where that started from and where it ended, I don't remember directly, but a 2-line improvement was the best improvement we saw.
Thank you. That was very interesting. And as an engineer, I think I understood it. But my question is, when you do the gene therapy, let's take a particular rod or a cone, and you've got the gene in there and you've corrected it. Now that rod or cone will replenish itself over time. Don't the cells make a new rod or cone? And does that keep the stuff in there?
No, so these are postmitotic neurons. So they grow, they form, they stay there, and there's only one of them. And if they die, they're gone forever.
Thank you.
Sure.
What is the frequency of this particular condition, this gene?
It's a fairly rare condition. It affects about 3,000 patients in the US and the EU.
Okay. And how do you even find them then to do your study? That's
a good question. So with all the work that Bill Houseworth did and his collaborators on LCA2, which ultimately led to approval of Luxturna, there's become kind of a heightened awareness in the major markets that treatments for these rare diseases are up and coming. And so there's many centers of excellence around the country now that focus on genotyping their inherited retinal disease patients, because they know now that it's no longer that the doctor would say there's nothing you can do, go learn Braille, get a cane, get a guide dog. There are actually treatments being developed. So there's increased awareness, and we've really benefited from all of that and have been able to find a lot of patients actually. And I think a lot of these are actually underdiagnosed, because a lot of, kind of routine, you go to your optometrist, they're not going to suggest genotyping, right? You only get that if you go to a specialist.
Is there any chance that anything along this line can be used for macular degeneration, like an anti-VEGF
Yeah, there's
growth factor, monoclonal antibodies, turning those genes on?
Yeah, so you probably know the standard of care for AMD, if you have the wet form, which means you're making new blood vessels, is to go and get an intravitreal injection in the front of your eye of an anti-VEGF antibody. Those go by various names depending on the company, Lucentis, Eylea, et cetera. But companies are going after a gene therapy to deliver that same antibody fragment so that they can get one injection and then never have to go to the clinic and get another shot again. There's, that's still being tested, and it's a lot more difficult to do gene therapy in the front of the eye than it is in the back of the eye, because even though the eye is immune privileged, the subretinal space is the most immune privileged. The vitreous is less immune privileged, and the anterior chamber is even less immune privileged. So the closer you are to the peripheral circulation and like leakage out of the eye, the testier things get. But there are a lot of companies pursuing a gene therapy for that, because that's a large market indication, and you know what big pharma cares about, right? Yeah.
Sorry.
A number of years ago, I heard a lecture at Whitney, and it was involved, the lecturer spoke of blindness and a study involved with blindness because of, I think, a lack of vitamin A. Did you know anything about that?
So there were physicians that used to suggest taking vitamin A if you had certain types of retinitis pigmentosa. I don't know exactly what lecture you're referring to, but yeah, I don't know. I would have to think about that. You don't remember the person's name?
No, it was a number of years ago. And
Yeah.
also I'd like to know when you were actually at Whitney.
Okay. I was there the summer of, God, when was I? I graduated college.
Come on, Shannon, you're not that old.
2000. I was there in 2000.
2000. Jane.
First of all, there's a chat from Massimo Downey. Don't some eye ailments show up as adults, and do they have a genetic basis?
You want me to do that one first? Okay. Yeah, definitely. There are diseases like Leber's hereditary optic neuropathy that can show up later in life. It's a ganglion cell dysfunction. So patients actually, they go blind very, very suddenly. So there are definitely conditions that present later in life. Usher's syndrome, certain forms, milder forms, the patients go blind, in their third or their fourth decade. So not all of these inherited retinal diseases present in infancy.
And my question is just a clarification. So you lift up the layer that has all the neural stuff in it, all the neurons in it, and underneath are the rods and cones, and you're just sort of squirting this stuff in extracellularly.
Yes.
And it manages with the virus, et cetera, it manages to just get itself into those cells.
Creating a puddle of fluid inside a virtual space that you've created with that needle. And then you're getting the virus direct access to those cells, and it immediately infects them. And the retina lays back down too within hours of the surgery.
I presume this is all FDA approved now, since you're
No, so
using it in patients?
So no, we got our IND approved, which is the FDA's allowance of us to start a clinical trial, but to get the gene therapy approved involves a phase 3 trial and a BLA.
So you lift up, like Jane was asking, and in a small place with a small needle, you put some of the genes that you, or the AAV, does that go all the way around the back of the eye to get to everywhere, or do you have to do it in several places?
That's a great question. So with early generation AAVs, I should back up. AAV comes in a variety of flavors. There's not just one AAV, and Arun specializes in making new flavors of AAV. But with the earlier generations of AAV, you would place it in a puddle, and that would only detach a very small portion of the retina. So you're exactly right. Like if I wanted to target genes to the foveal cones, I'd have to detach the fovea because I could only infect cells that were in that little puddle that I created. And that's interesting because sometimes patients don't respond well to having their foveas detached with a subretinal injection. The peripheral retina seems to handle it okay, but the fovea doesn't. So one of the things that I did in my lab, this is like the perfect segue. Who invited you? One of the things I've developed in my lab is an AAV that can spread beyond those margins so that if we want to get genes safely to the foveal cones without having to detach that retinal region, we can inject it further off into the periphery, and by virtue of its spreading ability, it gets safely into those cones and infects the larger region of the retina. Sure. Thank you.
How many patients have you treated now? Showed that one little girl.
15.
15 patients, and it worked in every one of them long term.
We had one that developed a bacterial infection. As I mentioned, it was a complication of surgery. So in that patient, we couldn't say much, unfortunately, but in the vast majority of patients, yes.
Yeah, okay.
Mhm.
I assume you start with one eye, and then when you treat the second eye, do you get different types of results? Like, is it faster or is it slower?
Yeah, that's a good question. So in a phase 1/2 trial, we always start with one eye only, and we purposely do the worst scene of the two eyes, just in case something were to go wrong. Once you prove that it's safe in your phase 1/2, then in your phase 3 trial design, you can do bilateral treatments. And the timing, this has only been looked at once because there's only one approved retinal gene therapy right now. But the timing, the onset of therapeutic effect was similar in the second eye as to what happened in the first eye.
This is any other questions on Zoom? No. Okay. This is amazing. Your accomplishments are really fantastic. Thank you.
Thanks for the invitation.
Where do you go from here?
Where do
What's
I go?
next for you?
Phase 3, and at Cena Therapeutics, my company is working on another phase 1/2 clinical trial for another inherited retinal disease, which is also working.
A different gene.
Yeah, I'm going to do all the genes.
Yeah, you're doing that. Okay. Thank you so much for coming and presenting this. This was fantastic. Next week, our speaker will be speaking on